So as we know, for COVID-19, the role of adaptive T-cell immune responses in disease pathogenesis and the longer-term protective immunity is currently poorly defined, but it's essential to inform potential therapeutic intervention and enhance the vaccine design. Most vaccines are aimed to induce humoral immunity to the spike protein, but it is not yet known if this will be sufficient to induce full protective immunity to SARS-CoV-2. So studying the natural immunity to the virus, especially the role of SARS-CoV-2 specific T cells, therefore it's critical to fill the current knowledge gap for improved vaccine design. We recruited 42 individuals following recovery from COVID-19 in Oxford, including 28 mild cases and 14 severe cases. Actually, peptide pentamer was synthesized by Pro5 comprising 5 peptides bound to the appropriate class 1 molecules we defined. And the right panel shows the example of pentamer staining. And strikingly, and despite almost half of the individuals possess A2, we were not able to identify any dominant A2 epitopes. Pentamer folded with A2 with the spike epitope reported previously by other groups was also synthesized by the Pro5, and we only see 1 out of 6 A2 positive donors tested, and which shows very weak responses and with very low frequency. So the ex vivo tetramer staining also showed typical central memory and effector memory and the early and or intermediate differentiation phenotype of SARS-CoV-2 specific T cells in recovered patients. So in summary, we found robust memory T cell responses specific for SARS-CoV-2 in the blood of donors who have recovered from COVID-19. The breadth, magnitude, and frequency of the memory T cell responses was significantly higher in severe compared to mild COVID-19 cases. The T cell responses correlated with antibody towards the spike and RBD, and a particular high frequency of spike protein specific CD4 T cell responses were observed in patients who had recovered from COVID-19. And furthermore, we identified epitope clusters and target by T cell across the donors, and a high proportion of SARS-CoV-2 specific CD4 to CD8 to CD4 T cell responses is also proposed in the mild cases.